you. Again, this is an honor to present some of our results, and this is only an experimental situation and preclinical studies. These are routines that this is my view, and what we are thinking that we are working on nano delivery of drugs, and in this case we are also using nano delivery of stem cells. These are experimental situations. I'm not very expert in stem cells, but we did some experiments in animal model. It's one technical innovation that if we combine with cerebralizing nanowire stem cells, I'm showing some example of this. So, this was the experiment in closed head injury and in different environmental conditions. Of course, this head injury is important in many aspects in clinical situations, particularly post-traumatic stress disorders. And these are also significant stress that they produce. These are some situations you can see, and since we are working with military organizations, this is important, and need to know that most of the head injuries are due to firearms, and concussive head injury is very well known and described. So what is happening that when there is injury on the head and skull is not perforated, then we have counter to injury, and that is more dangerous than under normal circumstances. We have a model in animal experiments in which we apply a weight that is dropped from 20 centimeter on the skull, produces very similar counter-coup mechanisms, and I showed yesterday the results that the uninjured side is much more damaged than the impact side, and that is quite natural. And here is the condition at the electron microscopy. The left side shows much more damage than the right side. And then we tried this work with mesenchymal cells and a combination of cerebralizing. Nowadays, there is much more of experiments going on either silicon nanowires and copper nanowires. The stem cells grow better, and even people are talking about nanowiring of bacteria. We have very nice presentation by the Nobel laureate yesterday, and this is in the latest issue of Science in November. There is one report telling that neuropeptide treatment with cerebralizing improves the survival of neuronal stem cells. This is a work from the United States just published. So we are encouraged that probably we are also going in the right direction. I showed this diagram yesterday. It is a multimodal drug. It's a combination of different neurotrophic factors and active peptide fragments. And I'm happy that the previous presentation showed that these neurotrophic factors, if delivered using iron oxide nanoparticles, they are doing much better. We are very happy to know that. And this is an example that when we produce injury at 38 degrees Celsius as compared to 21, the parameters like Evans blue leakage, iodine, rainwater, brain swelling, and neuronal injuries are exacerbated as compared to the normal temperature. And you can see here that if we have given nanowired mesenchymal cells, they are significantly reduced, but a combination of cerebralizing and stem cells, they are even much more better under these circumstances. This is an example here. You can see that the combination with mesenchymal cells and cerebralizing shows many live neurons after traumatic injury at high ambient temperature as compared to the mesenchymal cells nanowired with cerebralizing alone. In this case, cerebralizing is not nanowired. So we can say that hot environment exacerbates brain pathology, And combination of mesenchymal cells, nanowired, together with nanowired cerebrolysine is better. And then I'm showing you some examples in hyperthermia, because if the results are universal, then they should also show similar kind of neuroprotection in other kinds of pathology. And here is also the example that WBH means whole body hyperthermia in diabetes produces many leakage to albumin positive cells as compared to the whole body hyperthermia alone. And when we have given this nanowired mesenchymal cells, we can see that the leakage of albumin is significantly reduced. The same thing is happening with neuronal cells. Many live cells can be seen when we have given nanowired mesenchymal cells in hyperthermia. And this is also seen here at the level of electron microscopy. We can see that myelin is better preserved. So in this case, we can see that not only traumatic brain injury, but even hyperthermia also, nanowired mesenchymal cells produce better results. Another example in final phase, we tried in experimental Alzheimer's disease model where amyloid beta protein was injected, and we have studied some pathological changes. This was presented in Society for Neuroscience this year in Chicago. And we have studied neprilysin. Neprilysin is shown that its level is reduced in Alzheimer's disease, and this is the recent trend that those drugs, if they can elevate neprilysin, may have better results. So we have used the same thing here, and you can see that when we have given nanowired mesenchymal cells with cerebrolysine, the level of neprilysin that is significantly decreased was increased here in the combination here in all cortex, hippocampus, cerebellum, thalamus, hypothalamus, and brainstem. So this shows that nanowired delivery of mesenchymal cells is superior than the stem cells alone. And the main points are given here. So we can say that if we deliver even stem cells using nanowired delivery, we have better results as we have seen earlier. And I stop here for your kind attention and some advice in this research. Thank you very much.